to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 15th episode of the Weekly Bioanalysis. My name is Dominic Moreno, and I'm the Senior Scientific Advisor for all things related to ligand binding assays, biomarkers, and flow cytometry. I've been with KCS for about 7.5 years now, and I've been in the bioanalytical space for close to 26 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, Senior Scientific Advisor with a focus on all things related to LC-MS technologies. I've been with KCS just shy of 10 months, but prior to that, I've actually spent 25 years in the bioanalytical industry, so I have a wealth of experience to pull from.
Today's podcast, as always, is brought to you by KCAS, Bioanalytical and Biomarker Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industry for over 40 years now. John and I are senior scientific advisors at KCAS, which we've mentioned many times now, and either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCS services. We are glad that you are tuning in to us today in the dog days of summer during a pandemic. As mentioned, this will be our 15th week recording the podcast. So if you're interested in the other episodes, they can be found at kcsbio.com. As a reminder, you can follow us at Twitter at The Weekly Bio and at KCS Bio. As we mentioned before, we started this whole thing because of social distancing. And this is a way of getting away from our normal mode of communication, which was face-to-face visits and going to see some of our clients. So even today, John is completely in a completely different place. I'm in Kansas City, and John is in upstate New York, and our producer is located in a different place in the Midwest. We are excited to share with you that we have had over 13,000 impressions. And as I mentioned last week, I want to clarify, it's almost 3,000 hours of time have been spent watching or listening to our podcast. The weekly podcast now gives us a chance to get to know all of you, and we hope for you to get a chance to know us. And even more importantly, it's a chance for you to get to know KCS and the services we provide. As always, the podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we will wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're very happy to have you here. We look forward to a fun and exciting episode today. To kick us off, John is going to go over today's podcast a bit more before we jump into the news and resources. John?
Yes, hi Dominic. So usual structure. We'll start with a list of the agenda items like I'm doing now. Then we'll go cover news and notes and resources, mainly focusing on COVID, but with some other non-COVID news as well. Then the agenda and the main portion is the purpose of validation, how it gives confidence in bioanalytical data. So have a good discussion on that. Then before we have feedback and questions, and then moving into a wrap-up with a teaser for next week.
All right. Well, what do you say, John? We'll just jump right into the news now. News and resources. You want to kick us off and tell us about what's happening at Lilly?
Yeah, let's go with that. So, Lilly have been very involved in the whole COVID-19 research, etc. We'll touch on them later in another item. But one of their antibodies, they've actually moved into phase 3 within nursing homes to see if it can be used as at least a temporary preventative against the virus. I mean, it looks like a really sort of exciting development in terms of a trial that's being conducted. So, what's your thoughts on that?
Yeah, I think it's clever that they're going into that setting. I think that's probably where we need it the most, right? That's probably the most vulnerable population for COVID. So that made a lot of sense. And then, there's such a big player in the field. I think it's excellent that they're getting involved, which in terms of the field itself, maybe that leads in nicely to the second segment, which is to me a historic thing to have. It came out that AbbVie, Amgen, and Takeda are in a joint COVID study, and that's an amazing. So to see all these big pharmas kind of rallying together and big biotechs like Amgen rallying together and getting together, damn it.
This is really interesting. I mean, I know you sent the link, but it's the first trial is three drug makers coming together, but it's actually part of a bigger R&D, the COVID R&D Alliance, which is in excess of 20 um, you know, biotechs and pharma companies, and this is so Lilly's one of them, but it's AbbVie, Alnilam, 58AM Ventures, Amgen, Arbutus Biopharma, AstraZeneca, Bayer, Bristol Myers Squibb, Eisai, Evotec, Gilead, GSK, J&J, Novartis, Pfizer, Roche, Sanofi, Takeda, Theravance, UCB, Versant, and VIR. So, all with this, intent of how can we bring things to market to or trial them to bring to market to address COVID. And the first three, so it's a fluid trial, but the first three working together are, like you say, Amgen, AbbVie, and Takeda, and it's looking at the different potential therapies of each company and how they can be possibly combined with remdesivir and dexamethasone as the only two approved anti-COVID compounds. And if they show in lack of efficacy, they'll maybe the trial can be molded to then add additional arms with additional investigative therapeutics. So it's all really an interesting model. But the fact that you've got all these pharma all working together is really the big deal here. It really is impressive.
Yeah, it's historic to see them, uh, 20 companies coming together to kind of tackle the problem here. And I really, when I hear these types of things, and I know this is in the therapeutic space, but we also know it's to some degree there's a lot of collaboration in the vaccine space. This does make me a believer that a vaccine could be ready. I still think it's aggressive to say by the end of the year, but it gives more hope and more, kind of scientific backing to sort of support the idea that we can expedite things if we're rallying together rather than in the past it had been, a company had to like tote the line by themselves, right? So
Exactly.
it's been a
Yeah.
really, it just it you hope that maybe as we move forward and out of COVID and we've solved COVID, maybe we can get on oncology this way and we can tackle Alzheimer's disease and dementia this way. And
Yeah.
hopefully it'll go because I think there's plenty of meat on the bone for these companies to be profitable. They don't all have to, do it alone.
I think that highlighting, the CNS type drugs, where you say if you could approach that way, that really would be impressive, because we've seen so many drugs, being tested for schizophrenia, for Alzheimer's, for all these mental health and degenerative diseases, but fall by the wayside. I mean, if you read the industry newsletters, there's regularly yet another drug drops out where like I say, it's a CNS drug and it just isn't addressing the condition at hand. So yeah, if you could translate from what we're doing from COVID to other therapeutic areas, that would be very impressive. So this clinical program is dubbed I-SPY, is basically looking at three different drugs to really try and deal with the life-threatening immune overreaction that we've seen in COVID-19 patients. So the anti-inflammatories, in its question, can each of them address that issue and hopefully give us a therapy to deal with that portion of the symptoms that some patients see.
Yeah, it's a clever name, the I Spy. I like that. Yeah, it's just again, I think as a scientist who's been doing this for such a long time, it's really comforting to know that we can get together and tackle such a massive issue. Maybe we'll move along to Dr. Fauci. He dubbed it insidious increase, and he's worried about COVID. And John, I got to tell you, before we got online, I heard you talking about U.S. not being able to travel to all about 9 countries, and I think that's the irony behind that. Maybe we can talk a little bit on it, but I do think that I don't know if we let our guard down a little bit, and this is causing it. And even as this, it's funny because even as this article that I'm reading was from like 3 days ago, you've seen some plateauing in states like Florida, Texas, and Arizona. They're actually starting to come back down. So, I don't. I certainly think that this is not the second wave. I feel like What's happening now is just we let our guard down. We're testing more. We did a whole bunch of things that, maybe we loosened up things a little bit, and that caused this massive spike we're seeing. But this isn't, to me, it's not the second wave. I mean, I think we should all be preparing ourselves for, sometime in the late October, November timeframe when we really start to see, virus season kind of hit. But I think it's, his this article's fairly interesting around, the words he used is insidious. So I don't know. It
I,
just, again,
I
it's tough
think
to discern what's happening because already some of what he's saying has to be backed off a little bit, but clearly there was a spike.
I think it's a yeah. I think where it was interesting to me is he basically says, there's five or six fundamental things that we can do to really prevent this spreading. And if you look at the list, it's correct wearing of masks, avoiding crowds, physical distancing, prioritizing outdoors over indoors, and following adequate hand hygiene. And the key one for me is he says staying away from places like bars where people congregate. That is
Yeah.
obviously the indoor over outdoor. And the UK has done the same thing where they've moved to open, and pubs became a priority to open. And then it's like apply common sense in the pub. And I'm sorry, I'm speaking for myself here. Get a couple of beers inside me, and common sense is out the window. And to me, a bar really, of all the environments to open up, bars make no sense to me. But equally, that's a big industry, so somehow you have to find a way of at least giving some sort of financial assistance to the industry to get it through this, because actually we don't really want bars open if we're going to get this under control. That, that
Yeah.
that's me on my soapbox here. But it is all simple. I mean, we talk about, here's what the pharma companies coming together to come up with cures and vaccines and everything, and then here's what we can do, and it's all very simple stuff. And it is a worry that, where we talk about rushing into getting bars open, schools might be another factor. I don't want to say too much on that, but it's a concern. When I think about my kids' school, which they've moved on from, if they open up the schools here, what sort of engineering controls can they put in place to keep people safe? And knowing the logistics and flow of the building, I don't know how they can do it. So it's a, yeah. That it was a good article. But like I say, there's simple stuff we can do to really help, and I do worry each time we sort of seemingly get it under control, or it's going down. It's like, oh, we've got it, we've solved it now. Let's
Yeah.
go. And you look at Europe and Asia and other countries, and even countries that I would consider to have it under control are still having flare ups because you just let your guard down a bit, and it can explode. Um, in many of those countries, they have a good test and trace approach, so at least they can tamp it back down. But it still shows that everyone's vulnerable here. I mean, certainly in Europe, they
Yeah.
saw quite big flare ups. But I do think they have the processes in place to sort of get it back under control. But it just shows you can't relax with this at all until we really have the therapies in place to deal with it.
Yeah, and along the lines of the bars, I think it should say like clubs. Those are really the
Mhm.
places where he said bars specifically, but most of the bars around here, and I haven't been to a whole lot of them, but when you walk in, they do have some restrictions that don't let you get too close to each other. No one's really going out, to be honest, so none of them are like packed. But it's the club scene that I think is where you're getting the probably the higher infection rate, and then just I'm just you mentioned, oh, if I had a couple of beers, I'm sure all of my social distancing kind of goes out the window. I could only imagine if I was in my early 20s and had a couple of beers in me, and then it's like forget social distancing, absolutely. So it's just something that we're, for the sake of time, we'll move on. And then you did mention the schooling, and we got a tough decision coming up September 7th. Right now, it's open here in Kansas City. I don't. I feel like in Johnson County where I am, the outbreaks are not. It's not bad. There's not a lot of cases. So I don't know, John, but there's no way they're going to be able to adhere to these things unless there's you stay in the classroom and the teacher rotates. But the moment you start rotating kids in hallways,
Yep.
who knows?
Yeah.
And as we said offline, I think we should, me personally, if I were running the country, I would say, look, we're going to try to open things up. And if we have to, we can walk it back. but everybody should be prepared to have like a 4-week shutdown or a 3-week shutdown come, you know, November 15th or something like that, because we really should be. I think if we could plan it, that would allow for industries to be prepared better, worrying about daycare and all the challenges that some of this brought on. If you know there's going to be something and we're just going to kind of get ahead of it, I think that would be better. But, and I don't even know if that's true, because during the warm time, we're seeing virus outbreaks, which is a little bit unique. So this is a challenging thing, but I think for time, we're going to have to
Yep.
let's move on.
We
Maybe
could
some
talk about it another
brighter
time.
news, right? Like,
As we get more developments, we can talk about it another time.
yeah.
So do
But there's
you
some
want to
good
go with the
news.
next
The
one?
FDA, right? Yeah, the FDA
Yeah,
the first authorized antibody COVID test that can quantify. That's big news. I mean, we've talked about a lot of the tests out there. I still think there are a lot of bad tests, but it was good to see that somebody's got something that they can give a quantitation, which will be, I think, very relevant in a number of ways to know how protected you might be or how infected you might be by
exactly.
your antibody levels.
No, that's certainly going to be one to watch. So yeah, very interesting to you start getting a better handle on how much antibody you have, and then maybe be able to correlate that to what it means in terms of protection, etc.
Yeah, it is definitely in the right direction, and I think there's been a lot of development in the antibody space that's gotten better, and we're hoping even here at KCS to be able to have an anti-COVID antibody screening, not an anti-COVID, excuse me, a COVID antibody screening test up and running here in time, because from a clinical trial standpoint, you can only envision that. Me personally, whether it's 6 months from now, 6 years from now, or whenever, I think you'd want to know if somebody's got copious amounts of COVID antibodies in there versus maybe just some circulating levels. So all
Exactly.
really,
Yeah.
really good stuff.
And then in parallel, you have on the other end of things, the NIH has picked 7 COVID-19 diagnostic tests in which is an interesting approach in like a Shark Tank like competition and unlocking funding for further development. And much of this is driving speed to hopefully give test results quicker, because we've heard stories of people waiting days for tests, and in those days, obviously, you're interacting with people. You may behave normally, you may not, but if you don't find out you're positive until days later after you've had
Mm.
your test, that you could have, who knows what could have happened in the interim. So yeah, obviously the quicker the better, and it's an interesting approach. The Shark Tank
Yeah.
investment.
I think it made, just some numbers at you, right? There was 650 applicants. They whittled it down to like 100 promising ones. And then over a week assessment, 31 made the cut, and then from there they whittled it down to 7. So that's like, again, very good movement. Right? Normally those things
Yep.
might have taken God knows how long it would have taken for that to happen. So, and then continuing, 3 of them are hopeful to have tests within a half hour. This is great stuff, John. This is the
Yeah.
stuff that
Yeah.
hopefully anybody listening can take some solace in. Like, yeah, we're making this progress, right? A lot of
Well,
good progress towards fighting
that's
this thing.
exactly it.
Yeah, we're
We're
not
not.
out of the woods. I think we're still going to be looking towards 2021 before we might go to a concert, right? We talked about social distancing. Like, I'd love to go see some live music, but I think we're probably not going to happen in 2020. But still, very, very good movement in the right direction.
It's very impressive how much effort and how focused the effort is in terms of trying to get us to really get this under control, and a little bit more, not quite so technological, but equally interesting, because there are other parts of the process that even if we have vaccines, etc., we still need the ability to distribute them. Was a story about Honeywell, who had come up with a new plastic as an alternative to glass. They were ready to launch, and then COVID hit, and suddenly it was like, well, they put the launch on hold. So they then, rather than sort of just shelving it, they actually pivoted and started to use this plastic to develop smaller smaller vials that could then be used possibly for vaccines or other medications, such that they're producing a product that is some properties similar to glass. It's lighter than glass, but equally it helps the supply chain. So because as part of if you read any literature, glass is actually in short supply at the moment, and certainly Corning had been incentivized to try and produce more. So to have another alternative that could actually help the supply chain is great. And again, I just like this because it's showing how many different companies are thinking about this, and they might not be directly involved with drug development, but if they apply their expertise, they can help us all move forward to help the case against COVID.
Yeah, and even further, Honeywell's like an American icon company, right? They're one of the most largest companies. They got their hands in many, many things. They do N95 masks. But to hear these like repurposing as they are, that to me sounds like jobs. And we've got such massive job loss. So hopefully these are the things that are going to help, repurpose the labor force so that we've got something to do. So it's a great story. I love hearing about companies like you said that are not necessarily biotech companies or pharma companies that are still making major impacts in solving problems as we combat COVID. So it's a great story. And again, I think it's just to me, it's all signs towards employment and what are people going to do, maybe post COVID or even during COVID. And this is, to me, it sounds like, Honeywell hopefully can either, they didn't talk about it in the article, but they're either going to have to hire some people or at least keep some people jobs on board because they're also part of the airline industry and clearly they're not producing that now, right? So just
Exactly.
great stuff there and really a good story. John, we got time for one more item, and that is going to be our July drug
Yeah,
approvals.
drug
You've been
approvals, which
managing this. Tell us about what happened in July.
so July still keeping drug approvals for 2020 still going at a pretty nice pace. So five new drugs approved in July, 4 small molecule, and then one humanized CD19 directed cytolytic monoclonal antibody snuck in the last day of the month. So at least you got something to talk about there.
Yeah.
Yeah, in terms of indications, 2 oncology, 1 CNS, 1 infectious disease, so an anti-HIV medication, and 1 insecticide for the treatment of head lice. So an interesting mix, but yeah, still showing that small molecule has a lot to contribute. Well, obviously there's always a lot of interest in the large molecule side of things.
Yeah, no, that was a great summary, John. I don't have much to add. Like you nailed it. You got 4 small molecules and 1 large, and a nice mix of disciplinaries that it's covering. And it's always makes me itch when I read about head lice, which one of the drugs is treating. That makes me of course, I don't have any hair, so it's not a big problem for me. But still,
But it is interesting. Unfortunately, I didn't save the graphic when I saw it. But actually, 2020 is on pace to do significantly better than most recent years in terms of drug approvals. So there were. I mean, there were. I can't remember. I think it's 2015 was maybe a good one. I can't remember, but even at this stage of the year, 2020 is way ahead of
yeah.
most recent years. So even though we're in the we're dealing with the virus, etc., etc. There is a lot coming through on the pharmaceutical front to hopefully help for other conditions. So yeah, interesting. I just wish I'd saved it.
Yeah, no, I agree. It's very counter to what we would think would be happening, but clearly we're on, I would have thought there might have been a lag at this point. I mean, we've been locked down since March and they're still pumping out 5 or 6 a month, which is great news. And I'd love to drill into that biologic that was approved because it's pretty neat what they did there to kind of engineer how they're going about it instead of your traditional monoclonals. But for the sake of time, John, we're gonna
Maybe
move on to our actual podcast.
maybe another time we'd look at what are the highlight approvals of the year so
Yeah.
far, and look at what are the interesting ones. I mean, they won't all be large molecule either.
Yeah, no,
Yeah,
that
let's
would be
move on.
interesting.
So let's move
Yeah.
on to the main topic. So we thought it would be good to talk about the purpose of validation, how it gives confidence in bioanalytical data. We talk a lot about development, we talk a lot about sample analysis, etc., but we haven't really covered validation yet in the podcast. So I'll start off with just why validate. So our process is very much a sequential. The bioanalytical process is very sequential. You're asked to develop an assay for a molecule or a large molecule, be it small or large. You basically develop an assay so you can what you think is reliably measure it. In the small molecule world, that's a case of you develop an extraction, develop chromatography to the and then you obviously have the mass spec as your detector, and you're looking to develop an assay that gives the sensitivity so you can measure that drug at levels you're interested in. And at the end of development, you think we have an assay here. You pass it on to validation. Validation is basically the process to show that developed assay is rugged enough to then be used in sample analysis. So there's a lot of tests that we go through, and we'll touch on these as we go forward.
Yeah,
I,
and
I know what we've many of us have done as we've been in small companies that have grown larger. You tend to find that the development team people have different mindsets in this, and development people who do development don't necessarily appreciate the rigor or want the what's the discipline of validation and sample analysis. Different minds do work in different better in different places. So at least what it really is important where a development scientist can say, I've come up with this assay. It really is important to test it to then be ready to do samples, because having a developed assay doesn't always mean it's ready to do samples. Sometimes there's it needs more than just getting an assay in place.
and if you develop it in your laboratory, that it's got to be developed somewhere else and then validated elsewhere, right? It's so there's typically a process. So when you might have it under wraps in your laboratory, but at some stage you got to hand that method off to another laboratory in order to later stages you tend to not do the testing unless of course you're a GLP lab, and that's a whole nother. Very few companies are actual GLP labs except for the larger ones, and even then they still tend to validate the method internally, and then they want to outsource it because they don't want their scientists running thousands of samples. That's not an efficient way to do things. I want to
Exactly.
take a small step back, right? So
Yep.
when you think about the process, right, in an ideal world, how it's going to work is when they have their pre-GMP drug product, right, a non-GLP drug product, or even say a GMP grade product. There's usually a non-GLP tox study done, and that's where you should be doing your method development, whether it's small or large. That's where the method should be developed. It's not something a lot of people come a little late to the game, and then it's rush, rush, rush. And now you're trying to develop a method in a couple months. Really, a true method development for a PK, I'm going to focus on that for now. It's a nice iterative approach where you're going to have the ability to do a non GLP method development and maybe some sort of non regulated fit for purpose validation or qualification to run those samples in the non-GLP setting. And then when you get into your GLP talks, the method can be refined in between, the time that you start to do the GLP tox studies, and then eventually validated in a GLP setting, and then testing your GLP samples. So that kind of sets the stage for why to me that I hope that sheds a light on why you validate it and kind of how
So
you do it.
yeah, so we'd be slightly different in small molecule because in discovery or non-GLP, we'll probably pull together an assay that isn't totally robust. It's good enough to just measure, samples for a small study. When you move to validation, you really want to, you may use that discovery assay as the basis for moving forward, but there's a strong chance that you may actually completely change the extraction to give something as more specific for the analyte in question. So although the chromatography will probably be the same, likely be the same, there's a good chance that the extraction will change.
Mm. Yeah, no, that makes sense. And I maybe I generalized it a little too much. I don't think it's too different on the large molecule setting. And then, of course, I'll take a moment of pause and talk about the ADA component of things, which is still, to me, despite 20 years of trying to do ADA assays, it's still like an evolving, changing thing in terms of when to validate it. And what's happening now is, typically people would invest in, some would invest in doing a validation in the preclinical setting. Some would choose not to do it at all. And then when it got into first in man, they'd have a non-regulated method or a qualified method to do that. And then you don't validate until you get into phase 3. Now the FDA is sort of pushing you to, when you get to phase 1, they'd like to see a validated method. And again, I think it just comes down to the size of your company and what your resources are, whether or not you validate at phase 1, because it's not a requirement yet, but it's like they want it, is how the language is now. The FDA would want a true validated method for your phase 1 study. So just a little sidebar there, John. Why don't we let's drill into the performance characteristics, right?
Sure.
So why don't you go ahead and jump in there and just drill down, and like, I think to me it's a pretty iterative approach, or at least not iterative isn't the right word, but I think it's a pretty standard approach to how you validate a method.
Yeah, so the key components for us with an assay, how we assess whether an assay is performing well, is initially precision and accuracy. And if you've got good precision and good accuracy, then chances are you're on the way to a strong assay. Accuracy is the close agreement to theoretical, and precision is how tightly results are clustered. So you can have good precision and poor accuracy, or good accuracy and poor precision, which basically says your assay is not under sufficient control. So, like I say, you really need both, and that's where we talk about the 15-20 rule, where we're talking precision and accuracy of within 15% over the curve and 20% at the LOQ, because at the LOQ you've got lower signal to noise, so the chances are the performance might be you may need slightly wider acceptance criteria. But of course, we're always looking to develop robust assays. So if we can develop better assays with better signal to noise, and actually, although you may have the 20% of the LOQ, you may never actually need it, because you're developing a strong assay that it will set you in good stead going forward. And this is what I talk about in the early days of bioanalytical, where you're learning things. Remember we had assays where you'd be talking signal to noise of 3-1, 5-1, and longer term, we just, because we knew that samples were important, we really start to say we want assays that have signal to noise at least 10-1 at the LOQ, so you can readily pick the signal to the LOQ, which is your lower limit of quantitation, back up from background, because a signal to noise is basically the strength of the signal versus the mean of the background. And so if you've got 10-1, there's very little doubt about how you're quantitating, in our case, a peak, and then basing your numbers on that.
Yeah, similar in our world, right? It's signal to noise ratio. 1 to 3 is a little low. I mean, I think we target like 1 to 10 as our preferred. You can always get a method that's perhaps a little bit less than that, but really you want some, I think of it as real estate or distance between your blank wells and your actual like experimental, or your lowest calibrator in our, lowest standard.
Yeah,
Yeah. So and then like when you go ahead.
yeah, sorry. For us, the with small molecules, if you're talking something with a very low molecular weight, that actually can be quite challenging. So when someone comes to us with a project, and when you look at what the structure of the molecule is, you're probably most concerned from an LCMS point of view. High molecular weight, because you don't know how it's going to fit in with the mass spectrometer, if it's even going to come in the range, or actually low molecular weight compounds can be a real struggle as well, because in terms of that's the area of the mass spectrum where you're actually going to have a lot of background interference. So it can be tough to come up with assays for very very low molecular weight compounds.
Yeah, and we have ours are the more challenging methods are ones that are anything that's sort of sticky, just like in your world. I'm sure it's the same way. And then multivalency, so bispecifics, or even things that have or quadrivalent quadrivalencies to them are incredibly challenging. And then there's always like anything endogenous, similar to your world, something that might a promiscuous thing, or perhaps a monoclonal antibody that binds to something that binds to multiple things. So, meaning it might bind to one protein specifically, but that protein has other things on it. That's where you can get some perturbation of your signal to noise ratio, which in turn can affect your accuracy and precision. And then I do want, like, I want to take a small moment and talk about ADA assays. They really don't have accuracy per se because there's no standards. This is one thing people probably don't know about ADA assays. You don't have a calibration curve on there. There's no standard curve on there. So basically, how it works is you have a negative control, and then you've got positive controls. And usually there's a high PC and a low PC, and then the negative pool. And then when you actually run a sample, if it falls within a certain range above a certain background, and there's cut point determinations that are based off of the plate, typically that's called a floating cut point. Although there are 4 different types of cut points, and maybe that's a whole nother podcast, right? But then it's just a matter of if it screens as a positive, so the result comes out positive or negative. So if it's above a certain threshold, it's positive. And if it comes out positive, you go on to the second tier, which is confirmatory testing. If it's negative, it's reported as a negative result. And that's why we really want to make sure we don't get a lot of false negatives. False positives you can live with because you go to the next tier, which is the confirmatory step. Basically, it's the same step as a screening assay, only you do the sample with and without the drug product in there, and that causes inhibition. So if the signal's knocked down, and usually it's somewhere between like a 50% reduction in signal, that's what it confirms as positive. So when a sample confirms as positive, you report it as positive. If it confirms negative, then you report it out as negative. If it confirms as positive, you report positive, but then you go to that titering step. And when you titer, all it is dilutions of the sample, 1 to 100, 1 to 1,000, 1 to 10,000, however you've done it. And then the actual data bit you report out is confirmed positive, titer of 1 to 10,000, or titer of 1 to 100. The 1 to
Mhm.
10,000 is going to be a higher abundancy or magnitude. That's the word that's used, a higher magnitude of the ADA. So walking down ADA road, you got a little education there, John. I hope it made sense what I said.
Let's move back to PKSs though. So we were talking real validation. So I mean, you set your signal to noise with the for the LOQ, and that's really your sensitivity to the assay. That's the lowest concentration that you can reliably quantitate. I'll touch on selectivity in a minute, but also important is range of quantitation. So for us, what you want to do is produce a calibration curve where you can reliably measure the concentration of samples over as wide a range as possible, because to try and try and minimize dilutions. Um, but what we find is there can be at least in LCMS you can have technical limitations based on the molecule. You can have carryover on the autosampler. That means you actually can only maybe go over two orders of magnitude. More typically, we aim for 3 orders of magnitude. So a typical calibration range would be 1 to 1,000 nanogram per mil. But so that then plays into selectivity. So you have your signal to noise, but you also want to know that there's no interferences from blanks or other matrix components. So that's actually something we will look at. For small molecule, you have internal standard. You want to know there's no interference from the internal standard to the analyte. So in terms of selectivity, you have a number of tests that you're looking to show that if you change conditions, you can still reliably measure the analyte of interest.
Yeah, quickly, because I think we're running short on time here, but very similar in our worlds, in terms of the large molecule space. That's where an ELISA gives you a certain range of quantitation that is maybe an order of 1 or 2 magnitude, but then the mesoscale expands it maybe 100 to 1,000 fold, which has a lot of um, benefits to expanding your range of quantitation. And then in terms of selectivity, I want to make sure the audience understands what selectivity is. So typically it's you take 20 individual lots of matrix, and that could be 20 different people's plasma, and then you got to spike in at the low QC level and spike in at the high QC level, and then they got to recover within 20%. So that can be challenging. A lot of times that's where sometimes you have to raise your lower limit of quantitation because selectivity doesn't pass. So again, some challenges in our world. I think it's similar. Does that happen in your world a
Yeah,
lot, John?
yeah, we would look at matrix effects, which would be looking at multiple lots to show that you can reliably quantitate at both ends of the calibration curve. We will also look at particularly in LCMS assays, lipemic, so that's a
Hmm.
high
Hmm.
fatty plasma, and my
Hemolytic.
mind has gone
Hemolytic is
blank.
the other
There
one.
you
Yep.
go. Yes,
I gotcha.
so it's basically testing to show that you can, if the blood is, your plasma is hemolyzed or the blood's hemolyzed, you can still reliably quantitate your analyte of interest.
Yeah.
Um,
Sorry.
so
Typically, a bigger problem in your world. We don't. We will sometimes test hemo. We can. I think we test it routinely, but we don't see it as a problem. It's much more. Both hemolytic and lipemic samples are more on the small molecule side of things. I feel like unless it's warranted, we test it on the large molecule side.
for LCMS side of things, it's a lot of it is a case of, can these other components of your matrix affect your extraction efficiency? That's where a lot of it comes from. And the other part of with small molecules, we have the internal standard, which as much as possible we want a stable label internal standard, because that is effectively a heavy version of the analyte of interest, and at least in LCMS, it's going to behave the same as the analyte of interest. So for us, a lot of the variation comes, in terms of chromatography. Yes, you're separating your analyte of interest from other components, but you're not looking at those other components when you're on the mass spectrometer. So you don't really know what's coming out and correlating with the peak of interest. So you want to do tests that ensure that even if you do have other components that correlate with your analyte of interest, they don't affect the quantitation.
Yeah. No. And that segues into the last topic we want to talk about, which is critical
I
reagents.
think, can we talk about
Yeah.
stability assessments first?
Sure. Go
Um,
ahead. We can take a moment to talk about stability. Absolutely. Those are very important things. You
yeah,
bet.
so we'll get away from the experiments that really affect how, there's validation experiments that we've talked about, to a large extent are monitoring how the assay is likely to perform if things change in terms of what the samples are, etc. But the other really important part of validation are the stability assessments, and these are assessments that we do that really help determine how samples are treated in the clinic, how we can store samples, and how long we can store samples before analysis. We will do solution stability, so you actually show that your analyte is stable in solution. Which, if you have standard reference standard, it tells you how long you can use it for before you have to get a fresh batch to work off. So for us, it's typical is benchtop. What we call benchtop stability. So that's basically assessing stability in matrix at room temperature. We'll do freeze thaw, which is the stability um, to see if the analyte is affected by being taken in and out of the freezer. So going from minus 70 to room temperature to minus 70 to room temperature to minus 70 to room temperature, and seeing if that has any effect. There's obviously storage stability, which, like say, it means that you can, if you're running a study, it gives you if you've got months of long-term stability, it gives you the ability to batch samples rather than having to run them immediately. And then we'll do whole blood stability, which is really there to say how quickly you have to get um, from the sample from a patient into a form where it can then be shipped to the bioanalytical lab.
Yeah, and we're almost identical. We don't do the whole blood stability on our side. John, I think for the sake of time, we're going to have to move on to our feedback and questions because we were a little long-winded today, John, but that's okay. We can always have another podcast to go over method validation if we like. Look, we're going to drive right into. We got some feedback. We got quite a few people feedback today, and some of them are some hard-hitting questions. Maybe I'll go first. Cole asks, was curious if either of you had heard about recent use of ultraviolet light being used in ceiling fans to kill airborne pathogens in living areas. Since this is a family-friendly podcast, I won't mention the name of the fan company pioneering this research, but I'd like to get a scientist's feedback on the potential here. So, John, I don't know. Ultraviolet light has always been something. When I worked at one of my previous companies, they used ultraviolet light to purify their water, which was an incredible sort of technology that they had. In addition to all your standard filters and everything else, but they were making reagents for instruments, and they used ultraviolet light. I think ultraviolet light is something you use in a biological safety cabinet. Even this week, my wife and I were talking about, could ultraviolet light be used for COVID? It's interesting it came up. So I mean,
Hmm.
in theory, I think it can. There's also people looking at filters in your furnace to help cut down on it. And as far as I'm concerned, anything you can do to cut down on the bolus of the virus entering you will be beneficial.
So on that ultraviolet, what is being done in terms of cleaning the labs at KCS? Because Min has instituted a number of different things in terms of, you know,
We're
cleaning
using
the lab
bleach
post
with
business.
electric spray, right? So there's an electrostatic spray. I don't
Ah,
know if you saw that video. We're doing that nightly where they showed, it was a really cool video where a normal spray might cover like half a light bulb, but an electrostatic will cover the whole thing because it kind of like it's using hydrophobic technology to get the Clorox and the cleaner into a more spreading into a further surface area around things. So we are
okay.
spraying nightly. Yes.
Okay, yeah, I wasn't. I knew it was something additional, but because I hadn't been to the lab since
Mhm.
March, I wasn't actually aware of what was involved there. Okay,
Yeah.
okay,
No, good stuff. And like I said, if all of it is I don't know whether the scientific backing might not be there, but the theory is there around ultralight and even filters.
sure, yep. So let's move on. You're probably going to answer this one too. Lewis asks, "What is the difference between the current coronavirus and the previously known coronaviruses?"
Yeah, this one is funny, John. I don't want to misspeak, but I'm going to try and answer it. And if I'm wrong, I'll just follow up next week. But my understanding is there's been some mutations within the virus itself that have allowed it to become more virulent. So we had an outbreak in 2008. A similar coronavirus. And if you look on a Lysol bottle, it says, kills coronavirus. But this particular virus, there's some parts of a particular strain, and that's why it's COVID-19 that have made it more virulent. I think I got it there, but if I have to research that further, I'll double check that. But there's been some mutations or even like blending
Yeah,
within.
yeah. So my take on I think coronavirus, it's the part of a family of viruses, and this has just got different, like you say, mutations, it changes in the sequence, etc. That basically, you know make this attack differently from other previous coronaviruses. But yeah, it's I think it just differentiated. The coronavirus differentiation is based on the actual, the structure of the 3D structure of the virus, and I think that's where it comes from. But yes, certainly we can look into that.
Yeah, it's
So
MERS. So
basically,
it's MERS. It's coming to me now. It's the Middle Eastern Respiratory Syncytial or whatever that stands for. But like the coronavirus family, you touched on that. The family of viruses is incredibly diverse. So
yep,
it's not like this is a static thing. When you talk about coronaviruses, we probably have characterized thousands, and there may be many more that we don't know about. Remember, we're only as powerful as the antibody, um, the primers that are used in a PCR setting, right? What you don't know can kill you, right? So that there's only so many primers that we've identified. Although I think with sequencing and things like that, we've probably got a good handle on the number of
yeah.
viruses. But then
So
what happens is, maybe somebody gets co-infected with two different coronaviruses, and boom, next thing you know, you've got a mutation and it skips. So, great question, boy. I hope they get
Very,
easier.
very quickly, just very quick, I'm going to say here's what it says on the Mayo Clinic. Basically, coronaviruses are a family of viruses that can cause illnesses such as the common cold, severe acute respiratory syndrome, that's SARS, and Middle East respiratory syndrome, MERS, and then obviously we have COVID-19 is in addition to that family.
Yeah, I think we kind of at least I hope we drill down deeper than that for the audience. But yeah, that sounds good. The last comment here is from Diane. Diana, excuse me. She says, "Enjoy the episodes with all the resurgence of with all the resurgence of hydroxychloroquine in the news due to the recent social media fiasco. I was hoping to hear a measured scientific take, but considering the nature of the politics surrounding the methods and presentation used, I suppose it was probably wise to avoid it. And John, I'm not touching that with a 10-foot pole."
Nah, me neither. I just we'll sit and watch, and if there's anything significant that comes out about hydroxychloroquine, then we can talk about it further. But right now, I'm done with it for now.
I think I can make one comment. I think it will go down as one of the most controversial drugs in the history of America. Let's just say that, right? Like nothing has been in my 30, 25 plus years of kind of being involved with drug development. This one, standalone, nothing has been more polarizing than this drug, and nothing has been more like, nope, it doesn't work. No, it's incredibly effective. It's just a mess. It's
Yeah,
just not good.
yeah, yeah, and yeah, let's leave it for now,
Yeah.
and it's one of those things. In the long term, we'll get a proper. I'm sure we'll get a proper verdict on it, but right now everyone's too close to it.
Correct. That was a good way to segue out. So, John, we're wrapping up podcast number 15 here. Any big plans this weekend?
I was talking to Jeremy about this beforehand. To be quite honest, it's going to be another typical COVID weekend of doing next to nothing, and I'm at the stage now where I really want to do something, but quite honestly don't know what to do with it within our current constraints. So I'm
Yeah.
a little bit of a bit of a downer on that one, I have to say.
Yeah.
Yeah,
Well,
I mean, I'm sure it'll be relaxing. The usual. We'll make some nice food. We'll drink some nice wine, and then we'll be hopefully ready for next week.
yeah, well, I think it's relaxing, but you better make sure you love the person you're partnered with during these times. I've said that from the onset, right? But for me, I was supposed to go to Boston this week, and like as of August 1st, the state of Massachusetts mandatory to COVID test within 3 days. Well, I thought about getting tested. They said 5 days before you can get results. I'm like, well, that's not going to help me. Then they're like, oh, well, you can quarantine for 14 days. Well, John, I'm well past the age where I'm going to lie on a governmental sheet on things. So not that I would ever have done that in my life, but I'm not going to fill something out and then not oblige by it. So I had to cancel my trip to go see my mother, who's celebrating her 75th birthday today. And then I had a niece who had a birthday. She turned 14 2 days ago. And then this weekend I turn, well, I'm going to leave that alone, but I'm late 40s. And my birthday's Saturday, and we're actually going to make a trip to my in-laws. So we're going to go to Nebraska, and there's actually a lot of delayed graduation parties that we're going to attend. One of my niece's cousins is having his graduation party this weekend, and there seems to be a lot of that. These poor high school seniors who had to delay it for months. A lot of them are having their high school graduation parties this time of year, which I think is great because you gotta, everybody wants a $25, some money in a card for them for their graduation, so to speak. So
Yeah,
that would
it
be my
definitely.
excitement. And then where I got so much vacation time now, John, because I haven't been able to use any of it. And we were thinking about maybe going to a cabin somewhere and trying to take we were looking at like Table Rock in Arkansas is a real nice place, and we saw this great resort, but we're not going to do that next week, in part because we're just the whole COVID spiking, and really like when we called them, nothing's open. They're like, yeah, you can come, but there's not much to do. So I don't know. I might be taking a staycation next week is what's going to happen to me. We're talking about my wife already has the week off. I'll probably take a few days off, and I certainly think I could use a couple days away from the computer. I think it's healthy, and we're talking about maybe going some local drives or going to some state parks and things that you can do that that are around you that you almost never do. But now we're going to find out some of the local scenery here. I think there's a hot springs we might go take a look at that's about an hour away. So similar to you, not nothing glamorous, right? Not going to
Yeah,
Paris like I
I
wish I
think
would.
I think we probably need to see if there's some things we can do. But I just read earlier today that we're up to 34 states now that if you come in from them, you have to do a 14-day quarantine
Yeah,
for New York. So it's definitely narrowing down our opportunities, or limiting our options as to what
yeah.
we might want to do. So we'll see. I
Well,
mean, like I say, like luckily we all get on together, so we can relax together, and there shouldn't be any major animosity or anything. But I'm sort of getting itchy feet where I really want to do something, and I'll touch on it probably more next week about the reason why. But yes, it would be nice to get out and about, but
yeah,
it will be
and
quiet.
you touched on why we wanted to go away. It's my 19th anniversary, wedding anniversary next week as well, and we were hoping to go to
Oh,
a nice resort. We
congratulations!
had one planned out, and that was part of our rationale. But now again, we're just cautious, and maybe we could be more risky, but it's just not worth it. So we'll have to push pause on that one. Although I do have some neat gifts for my wife, she'll have to like that. So
Has to.
with that, it was
Well, congratulations on 19 years, or
yeah.
commiserations, or which way, I don't know which way it should be.
Well, for her, I can't believe my wife is stuck with me. I mean, she's great. I'm the one that's doesn't give anyone a word in edgewise. John, you think you struggle? Imagine trying to have dinner with me every night for 19 years. No,
Is it like,
with
thank God
that,
he's got some food in his mouth, I can talk?
yeah. No, but that doesn't even work either. Nothing slows me down. All right, what a great that brings us to the end of the podcast. Thanks for listening, John. You got any last words for the audience?
Yeah, we just have to say what our topic is for next week. We are looking at talking about bioanalysis of tissues and other non-traditional matrices. So we'll touch on ocular fluids and CSF and some of the challenges that we have to deal with it. Actually, urine would be a good one to throw in there too because it has its own little quirks.
Yeah, definitely an area of expertise. Seeing as we're in the animal health corridor, we work in a whole variety of tissues. And then within the last few years, we've really specialized in ocular. Should be a great podcast next week. Excited to do number 16. Well, that will close out podcast number 15. Y'all have a great week.
Yep, thank you very much. Talk to you next week.